Hello, thank you. Thank you for being here. I'm really glad I made it. And I'm so happy to be here in Cuatro Ciénegas again. And I will just start my talk. So my name is Mirna Vazquez. I work at UNAM at the Marine Science Institute. And today I want to talk about microbial communities, of course, and mangrove restoration. So I want to take you into the microbial world of mangrove restoration. So mangroves are powerful ecosystems. They are great. And they are all over the world. They store carbon. They protect our coast and also connect the land and sea. And we are very lucky because in Mexico we are fortunate to have mangroves all over our coast, just right here. But not everything is just like nice. These ecosystems are vanishing, and are vanishing due to very different reasons. Among them is hydrological destruction and development, of course, like hotels and everything, and also climate change. Climate change. All these things have fragmented the environment and are causing that these mangroves environment are lost and dead, just like in this picture. But not everything is lost. There are a lot of efforts on trying to restore this environment. But the thing is that most of these restoration efforts focus on the trees and recovering also the water flow, but they don't look into the microbes. So what I'm here to tell you is maybe we can look to the microbes, and maybe if we look at these microbes, we can ask this question, like, can microbial communities help us to understand what is lost and also can be recovered in the mangrove ecosystem? And maybe more important, can they help us to design better restoration strategies? So that's the idea. Let's try to look into the microbes. So I will talk about many different things that I'm doing in my lab to try to understand or answer these questions. So I will start talking about how does microbial communities look like in an environment that is degrading, and also how does these communities look in the restoration efforts, how these communities change. Then we are going to compare these results with a global data set. And finally, we're going to talk about a unique system that we found here in Mexico that I think can give us a clue on how we can save these environments from climate change. Okay, so what happens when a mangrove is disrupted? So to answer this question, we went to a part in the Gulf of Mexico and took samples in 3 different areas. These areas were distributed based on the degree of degradation. So the fringe is the one that is less degraded and it's also very close to the water flow. And the next one is the basin, which is kind of degraded. And the last one is completely dead, like the ones that I showed you in the picture. So we took also sediment samples in different depths to see which were the important things for the microbial community. Like, for example, if the season, like the flood season or other parameters. So what we found is that the alpha diversity increases in the degraded zone. So in the dead zone, alpha diversity increases. So that was not expected at all. And also, we also found that the location is very important, meaning that each community is different in the 3 sites that we sampled, and has a modest but also clear effect. So the deeper layers are more similar among the different sites. In terms of taxonomy, we found interesting things too. Like, for example, in the fringe and the basin, which are the ones that are not highly impacted, we found bacteria like Vibrio and Marinomonas, also some Acetobacterium, which are bacteria that like these salty environments, so that's fine. But in the impaired zone, like the dead zone, we found other type of bacteria, like Chloroflexota, Nitrospira, Acetobacteria, and we also found a pattern related to the depth. In the surface, we found Bacteria, and in the deep layers, we found Bacteria and the Thermodesulfovibria, which are the ones that are here. I don't know if you can read it, but they are there. And so this was with 16S data, but we tried to use this tool that is called PyCross to try to infer the metabolic functions. And we try to contrast these two environments, like the one that is not degradated, the French, and the dead one, the impaired. And we found that, for example, methanogenesis happens in both ecosystems, but different types of methanogenesis happen in the French. Like, for example, acetoplastic methanogenesis is more common here, and in the impaired zone, methanogenesis based on CO2. Okay, so this is just like the baseline. How does microbial community look like in an environment that is degrading and the one that is not degrading? So, but also I was mentioning that we are making efforts to restore this environment. So the question then is, are we restoring these environments correctly? Like, are we actually recovering the same microbial communities? So to answer this question, we went also again to the Gulf of Mexico and also to the Caribbean to try to find these communities. So we went there to 3 different locations where there has been restoration projects. So the idea was to take samples on different stages of the restoration process, and also take some control samples, like a pristine environment and a dead environment. So the idea is very, I think, simple, just to see if we can recover the biogeochemical cycles and in these different stages of restoration, and if they actually resemble the pristine community or are something different. So that's the question. So we went there, we took the samples, and I wish I could show you the data, but they are right now sequencing, so it's not happening today. But I can just show you the physicochemical data that we have, like, for example, salinity, temperature, and things that we took when we were there sampling. And what we observed is that the degraded site is highly extreme. So they have high salinity, higher temperature, and they are more acid. See, this is like this red thing here. And we also found that there's like a lot of death-related environments. So it doesn't matter where you sample, all the conditions are similar regarding death. So this is interesting because probably this is defining the microbial community that we are going to find there. Yeah. So, okay, so this is Mexico. We are trying to understand the microbial communities in mangrove restoration systems and pristine in Mexico, but we want to compare this to what else, other places in the world. So for that, we are building this tool, which we call Magenta. And Magenta stands for Mangrove Gene Catalog. So the Mangrove Gene Catalog is actually, we are downloading and creating all the metagenomes that we find in the public databases, which are a lot, and are distributed in these, basically in these countries. And what we want to do with this information is to try to create a pipeline that we can standardize, like we can use all over, to actually be able to understand biogeographic patterns, like global patterns that we can see in the mangrove ecosystem, maybe identify some functional markers for degradation in some part of the world, or even detect endemics and ecological outliers. So this is Magenta. We're working on it, and yeah. Okay, so Magenta is like this global view, but local the stories we still need to do them. And I want to talk about this special place, which is in Tabasco. It's called San Pedro Martin. It's like 170 kilometers from the ocean. So this is an island mangrove, the inland mangrove. It means that it's inside the continent. This is weird, because the mangroves should be in the coast. This mangrove community is inside the continent. So we went there to try to see like which kind of microbes are there, and if they are helping the mangrove tree to stay there and not die or something. And yeah, so we went there. We took samples in San Pedro Martin, which is this point, green point here, and we compared this data to the ones that we had from Campeche and Laguna de Terminos and Celestún, to see if the microbial communities were different. And yeah, we found that in terms of diversity, all the coastal communities are more diverse, but yeah, the microbial communities from the inland mangrove are contrastly different from the ones that we found in the coast. Meaning that all these different communities are different in taxonomically speaking, We don't know if they are different in terms of function. That's a thing that I want to study. And this inland mangrove community is also different. It has a lot of endemisms. Well, I don't know if really they are endemisms, but there are a lot of unique SUV. So probably that could be interesting. When we look at the taxonomic profile, we also found that there's a pattern related to depth. So there are clearly some lineages that you can find in the top layers of the sediment core. And there are also some other lineages that are more abundant in the deeper layers. So this pattern is really clear in this sediment cores, and that's really interesting. And we also found some lineages like bacteria and modulobacteria. And if you look closer to the names that you can see here, you are not going to find any type of lineage like Vibrionaceae, Cygobacterium, Marinomonas, which are the ones that are just found in these salty environments. So it's a completely different microbial community. So I think this is very important, and I want to say that if we want to save an environment, I think this is some environment that maybe we want to keep, because it has a different community, and maybe we want to understand what is happening there. Okay, so how we use all this knowledge to actually intervene in the restoration process. I think that it's important to go back to isolates to actually being able to have an impact there. So we are doing that. We are now isolating microbes from the mangrove seeds and also from the sediment, especially nitrogen-fixing bacteria and also plant-associated bacteria. And I think this bacteria then we can use it to try to build consortia that we can use in the restoration process, and maybe it works. So, okay, so the take-home messages that I want to say. Microbial communities are sensitive indicators of restoration. And also, the storage zones, like the dead zones, have unexpectedly high diversity. So that's interesting. And the other thing that I want to say is that restoring zones help us to understand how community resemble, how does these biogeochemical cycles actually restart activating. And also the inland mangroves, we have to keep studying them because they are like a reference point of how these communities can survive a global change that is happening now. So, yeah, to truly restore mangroves, we have to look at the dimension of microbes, and this is all I have. Thank you very much to all my students and all my collaborators. They are not all here, but there are many. But thank you.
Mirna promised to be quick, and she definitely was. And that was good because Hector is nervous about like, oh, Gabriela is, launch is right, it's going to be there, and not just that, with a poster session. So we want people that after launch, those that have a poster, the posters are going to be right at the hotel. I'll be there and show you exactly in what room. So we will have the lamparas, and you can install your posters for the afternoon. And I do want you to be all the time because we have a great talk by Daniel Carrizo, and so I hope that I hope that they serve quickly our lunch, and we can do that. And who was raising their hands?
Hi, Mario Guevara from UNAM. I am just curious about which are the mangrove species that exist in the study sites, and if you were able to differentiate microbial communities across the species. Thanks.
Yeah, that's an interesting question. All these mangroves that we are sampling are Rhizophora mangli. We are not sampling other type of mangroves because, yeah, right now we are not trying to see the difference. There are groups that are working on the difference between the different species, and they have found some. So, but yeah, this is only Rhizophora.
Thank you for the talk. Very nice.
I have a question related to the species of the mangrove. Do you know if there is some works related with the genome of the species and there are some differentiation between them?
Yeah, there are a lot of works related to the genome of the species. It's interesting because it's not a monophyletic group, so there are different groups in mangrove, yeah, and that's interesting. And I don't remember well if they have found this convergence evolution. I have to check, but there are works. And also there are a lot of work being done right now at UNAM too about the population genomics on the tree, and they have found a lot of groups within Rhizophora mangre at least.
Yeah, because I was thinking that maybe some adaptation process could be linked with the microbiome or something like that.
Yeah, that's true, but yeah.
Last question, and then we'll have we'll match see who runs faster. And we'll have a prize for those that get on time.
Very fast. Thank you. I saw in the picture of magenta where you show the global map that in Africa there are no studies, or it was white. And I was wondering if it's because in Africa there are no mangroves, or if it's because of the reason, maybe they don't research there. Or...
Yeah, I think, well, we have been making filters on this database, and I can't remember they are, but most of the works that are in Africa are like amplicon sequencing, so we are discarding those experiments. That's why it looks, but definitely there's not much studies there. So I don't know, maybe it's because it's Africa. No sé. Yeah.
Thank you very much, Mirna.